Hey, I just wanted to quickly plug that if you guys want any tutoring, some new slots have opened. If you look in the description, there's a link to my physics and maths tutor profile in which I do tutoring on there. Hello everybody, welcome back to another episode of the A-Level Cookbook. Today we're doing the AQA A-Level Biology Paper 3 from June 2018. So this is the exact paper that I did when I did my A-Levels. So broken bones are repaired by cells called osteoblasts and osteoclasts. Osteoblasts secrete a hormone called osteocalcin in its inactive form. Osteocalcin is a protein, and the active form of this binds to a receptor on beta cells in the pancreas, stimulating them to release insulin. Osteoblasts have receptors for insulin. So here, this figure shows how the production of osteocalcin by osteoblasts is controlled by positive feedback. So positive feedback, remember, is where you have a deviation from your norm and then it goes even further and amplifies it, whereas negative brings it back down to a normal, right? So let's have a look at this figure and make sense of what's happening. So if we have our beta cell, which releases insulin, which stimulates osteoblasts, these osteoblasts release osteocalcin in its inactive form, and then an acidic pH changes this osteocalcin to become active because these osteoclasts release acid, right? So acid from these osteoclasts makes this become active. As a result, you get active osteoclastin, which then stimulates the beta cells to make more insulin, which stimulates osteoblasts, which makes more of this, makes more of this. You can see how it's a self-perpetuating cycle. So in this case, it says use the secretion of this is controlled by positive feedback. Use information to explain why. Well, first of all, you can see from what I was just saying before, that entire thing, you can see that eventually osteocalcin causes more insulin release because eventually all of that keeps happening. So osteocalcin stimulates beta cells to release more insulin, and this leads to more osteocalcin release. And it's a self-perpetuating cycle. Now it says the acidic pH caused by osteoclasts caused the inactive form of this protein to change into its active form. Suggest how. So use what the questions told you. Osteoclast releases acid, acidic pH makes it suddenly work. Now it's told you that in the question above, osteoclastin is a protein. So for some reason, giving it an acidic pH means all of a sudden it's able to work. So in this case, what it is that this pH is causing it to change shape. So what's happening is the pH causes a change. We need to be specific here as well in its tertiary structure, not active site, not in 3D. You have to say tertiary structure, right? We need to say how. By breaking or changing hydrogen and ionic bonds. Nothing to do with disulfide bridges. You can't just go ahead and say enzyme active site and all of that stuff because it's not talking about that. This isn't an enzyme. So then it says binding of insulin leads to an increase in the rate of respiration in cells, such as osteoblasts. Explain how. So you need to know about how what they're saying is this binds to the cells and all of a sudden you get an increase in the rate of respiration. So insulin, remember, is all about shoving glucose into cells. So the way it works is it will bind to a receptor like that, right? And then what happens is you get these GLUT4 channel proteins carried in vesicles, they fuse with the membrane, and they make a nice opening for the glucose to enter via facilitated diffusion. Once glucose gets into the cell, because remember it's saying binding insulin leads to increased respiration, we've now got glucose inside the cell, which is used in respiration, so you get more respiration. So that's exactly what you're going to say. So insulin, causes the fusion of vesicles containing glucose channel proteins, or GLUT4, with the cell surface membrane. Therefore, more enters these cells via facilitated diffusion. I'm going to write FAGDIF just for short. So then there's more glucose for respiration. There you go. You've got to work with what the question's telling you here. So chronic lymphocytic leukemia is a cancer that affects some B cells of a person's immune system. Rituximab is a drug that's used to treat CLL. It binds to a protein called CD20 on the surface of B cells, and if enough rituximab binds to a B cell, it can kill the cell. Rituximab kills both healthy and cancerous B cells, and then the body makes new B cells. So the amount of CD20 on the surface of these varies from one person to another. Doctors investigate the relationship between the amount of CD20 on B cells of a patient and the percentage of B cells destroyed by rituximab, and here's their results. Each crosses one patient. So what we need to do is we need to look at our independent and our dependent variables. So, and here, our independent is the CD20 on a patient, right? So they're changing the patient and they're looking at the amount of the CD20. And then here, the dependent is the percentage of cells destroyed, B cells destroyed by rituximab. This will come in useful. So then it says, what statistical test could the scientists have used to determine whether there was a significant relationship between the amount of CD20 on the surface of B cells and the percentage of B cells destroyed by rituximab? Give a reason for your answer. So we're looking to see if there is a link between one thing and another, as in a correlation. So what you're going to do is you're going to work out your correlation coefficient. You need to explain why. It's because we're looking for a relationship between two variables. We need to say what they are. So looking for a relationship between two, actually you can say two variables. Yeah, two variables. It'd be helpful to list them out just to be safe, but yeah, that's what you're doing. If you're looking at the difference in means between two things, so mean 1 and mean 2, then it's going to be, the p-test is looking to see if there's a difference between these statistically significant difference, right? That's going to be t-test. And if you're looking for a difference in the expected and observed results, that's chi-squared. Because we're looking at whether there's two things correlating and they're related, then we're going to be using a correlation coefficient. So now it says, from these data, what can you conclude about the effectiveness of rituximab in treating people with CLL? Do not include considerations of statistical analyses in this. So what we need to do is we've got to this is essentially an evaluate question. We need to say supportive statements for treating patients with CLL. This is what the conclusion is that they're trying to get you to look at. We need to look at contradicting statements, as in what data goes against saying this is good, and then validity as well. So can I take this and apply it to everyone in the universe? Is my research actually answering the right question? If we wanted to look at this perfectly, we would use rituximab and see how many cancer cells die. But what we're doing is we're looking at how rituximab the amount of CD20 and using rituximab to see how many B cells are destroyed by the rituximab, but it's told you in the question it's healthy and cancerous. So this doesn't directly answer the question, which is a point we can bring up, right? Now, as well as that, the validity is to do with how they did the test. So for example, if I told you I have 10 people in a room, I've done this example, I know, a million times, but I had 10 people in a room, I give them chocolate and they all shit themselves, and I say, okay, in conclusion, 100% of people that eat chocolate will shit themselves. You're going to be like, well, that don't make sense. And then I'm going to be like, okay, well, evaluate it. Supportive statements, 100% of people shat themselves. Fine. In my study, 10 out of 10 did. Contradicting, there is no contradicting point according to my data. However, validity is where all the problems come up. Because one, I'm saying that everyone shit themselves because they ate the chocolate. I don't know what type of chocolate they gave, what was in it, what ingredients, whether people had allergies to it, how I selected 10 people. There was no control to see whether the shitting was actually due to the chocolate. Because the control group should be what. So a lot of people say a control to allow comparison. I think that's useless because obviously you're comparing. Every group you're comparing. You need to say why you use a control. The reason we're using a control is to see that whatever we're changing is the actual effect. So what I mean by that is in my control, you wouldn't give them chocolate, and the other one will have chocolate. So then that means anyone who shits themselves was probably due to the chocolate. If I have no control, I don't know whether they all just had diarrhea or like gastroenteritis that day. And as well as that, the sample size is small and all of those issues that come into play. So if we look here then, what are we saying here about rituximab? So percentage of cells destroyed by rituximab seems to get higher as you have more CD20, right? Generally speaking. So that's a supportive statement. So a supportive statement is that more CD20 on cells leads to more percentage destroyed with rituximab. Now what we need to do is we need to go hunt down a directly contradicting statement, as in something that goes against saying that rituximab is good for this. So, I mean, immediately we can see here that, like, you go up and you've got some separate points here. So if you do a line of best fit, you've got some there above and below. So the range is quite high at them. And at the absolute best, it can only get up to about 80%-ish, right? So that's a contradicting statement because, you know, like it's saying, is it good at treating CLL, as in curing cancer? Well, we can only get up to 80%. So the max max destroyed is only 80%, right? And as well as that, that must mean it doesn't really cure cancer, does it? Because it won't kill all the cells. Now, issues with validity, let's have a look what's going on. So issues with validity here is, well, I mean, blah, blah, blah. We don't know about sample size and stuff, right? We just know that there are some, whatever. So we can't really go on ahead and say like a small sample size, really. However, we, again, like I said before, we're measuring the number of B cells, that percentage of B cells destroyed by rituximab, and using that to see how well it can treat cancer. But the question's told you rituximab kills both healthy and cancerous cells. We don't know how many of these cells are actually cancerous or whether they're healthy. So, you know, that's one of them. Don't know the proportion of cancerous cells killed versus healthy. What else are there? Problems with? Well, if you have a look here, this line is like practically the same. So around here to here, there's barely any effect. actually that's an extra contradicting point, is that below a CD20 of, what's that going to be? What value? Like, 4, there's very little effect. That's a contradicting point. And that's about it. That's all you can really say. There you go. So around 11% of people with CLL have mutations of a gene called Notch1. This leads to the production of a non-functional transcription factor associated with CD20. The doctors determined the median percentage of B cells destroyed by rituximab with people in CLL who had the Notch1 mutation versus those that did not. So again, let's have a look at our new independent and dependent variable, because again, that helps us make sense of what's going on. Independent is what you're changing, dependent is what you're measuring, right? So here, our independent variable is either you have Notch or no Notch. Notch1, yes or no, that's it. And then here, the dependent is the median percentage of B cells destroyed, right? It's really important that you don't just write what they're saying here, like, the doctors looked at the percentage of cells destroyed by this. Okay, actually, it's kind of direct here, but sometimes it'll say, like, I don't know, a student looked at the rate of respiration. You can't say rate of respiration. You're going to write that, you need to write the dependent as in what they're measuring as a surrogate of it, because this will be useful for later. Now, it says here, human blood contains approximately this many B cells per decimeter cubed. Use the median values to calculate the difference between the number of B cells per decimeter cubed treated with rituximab with this Notch1 mutation and those without. So this is the median that were destroyed, right? So the question's asking how many were treated with rituximab with this and without that was destroyed. Because so what we're going to do is we're going to work out, first of all, we know that it's around this, but they've told you the percentage that were destroyed by rituximab is 4. So you're going to work out 4 over 100 times 1 times 10 to the 9, right? So that's with Notch1. And then this is without, so that's going to be 22 over 100 times 1 times 10 to the 9. If we do that, we get 4 times 10 to the 7, and then for the other one, it's 2.2 times 10 to the 8, right? So it's saying use these to calculate the difference between the number of them. So you're just going to do 2.2 times 10 to the 8 minus 4 times 10 to the 7. If we do that, we get 1.8 times 10 to the 8. There you go. So then it says use all this information to suggest how the mutation of the Notch1 led to the difference in the percentage of B cells destroyed by rituximab. So I'm calling in now, a lot of people must have been talking about survival and reproduction, and unfortunately that was what was the case. Read the question carefully. What has it said? It said that Notch1 mutation led to the difference in the percentage destroyed, right? So then we need to go and look at, again, just real quick, what's happening. So again, if you have this mutation, for some reason, there's less cells that are destroyed, whereas without it, more cells are destroyed, okay? So then again, Notch1 mutation means that rather than saying difference in them, it meant that fewer were destroyed. Now go back to what the Notch1 mutation is. This Notch1 leads to a non-functional transcription factor associated with CD20. So Notch1, what it does is it makes a non-working transcription factor, which for some reason means less is destroyed by rituximab, right? And the questions told you rituximab binds to CD20. And the questions also told you that this transcription factor is associated with CD20 production. Now all the pieces of the puzzle are there to explain why. Because what's happened is a mutation, remember, is a change in a base sequence, right? So that means it's going to change the primary structure of the transcription factor, which then changes its tertiary structure, right? So then transcription factors, remember, what they'll do is they'll bind to the start of a gene, and then it will lead to RNA polymerase making mRNA, and then eventually gets translated, because transcription translation, right? So then it means this transcription factor cannot bind to the CD20 gene, so then you get less transcription of CD20 mRNA, so then you have less CD20. Because there's less CD20, there's going to be less binding of rituximab to the B cells, so fewer are destroyed. There you go. So in women, the first division of meiosis produces one daughter cell that has almost all of the cytoplasm. The other daughter cells consist of a nucleus surrounded by a very small amount of cytoplasm and a cell surface membrane. This very small daughter cell is called a polar body. Polar bodies do not usually divide. The same process occurs within the second division of meiosis, resulting in one egg cell and 2 polar bodies. The diagram shows the formation of an egg cell and 2 polar bodies during meiosis, and it also shows what happens to one pair of homologous chromosomes. This pair carries 2 alleles of gene A. So homologous chromosome, right? Because everyone loves to get this mixed up. You're chilling, you're diploid, right? Because you have one chromosome from mum and one from dad. One maternal, one paternal. These are homologous chromosomes because they're homologous of the same thing. So if I have, this is me with my right hand, this is you with your right hand. Our right hands are homologous because they're both right hands. It's just my right hand versus yours. So these are homologous because they're the same chromosome. It's just one's from mum, one's from dad. So whenever you're replicating your DNA, you're making these sister chromatids like this. This is a maternal, and paternal. They're copies of that same chromosome. People make the mistake of saying it's this, maternal. That's wrong. That's objectively wrong. These are just copies of the chromatids that came from this. Right, so maternal, paternal. In meiosis 1, the homologous chromosomes separate, as in these separate. So now you end up with a cell that has this and this. It's haploid now because it doesn't have maternal and paternal. It only has maternal. It's just got 2 times the haploid number, not 2n. And this will have the paternals. In meiosis 2, these chromatids separate now, so you get maternal, paternal. That's what happens in your haploid now. So it's really important that you just get that distinction clear, because people just love yapping about random things to do with homologous chromosomes, not having a clue what it means because they're trying to memorize answers and it don't work. So anyway, it says here, complete figure 8 by putting a big A or a little a in the boxes. One box has been completed for you with big A. So if we have a look, we have big A, little a, right? So if you have a big A here and a big A here, there's a little a here, then that has to also be a little a. You can't have any other letter there. Then it says, put a tick in the box next to the name of the process that produced the combination of alleles on the chromosome in the first polar body in this, right? So if you have a look at this first polar body here, this is big A, little a, right? But remember how I said the homologous pairs separate like this? It should be little a. So why on earth is it big A, little a? What must have happened is they must have exchanged with each other. So that's called crossing over. So that's what happens in meiosis 1. Right before the split happens, they cross over and exchange alleles. So literally, it's like this, right? And they exchange bits and bobs. Otherwise, you would just have the exact same gametes each time, and you would have the same, siblings. They'd be identical. So that means crossing over is that. A scientist measured the diameter of a polar body and the diameter of a nucleus inside it. The diameter of the polar body was this, and the diameter of the nucleus was that. So polar body diameter is going to be 10.4 micrometers, and then the diameter of the nucleus is 7.0 micrometers. The density of the mitochondria in the cytoplasm of the polar body outside the nucleus was this. So the mitochondria outside nucleus is 0.08 per micrometer cubed. It's saying work out the number of mitochondria in the polar body. You should assume polar body is a nuclear and spherical, and it's told you the formula for a sphere, and you should use the 3.14 they gave you. What we need to do is we've got to work out the volume of the polar body and then take out the volume of the nucleus. All the volume that's left is going to be how many mitochondria you can squeeze into this. So to work out the polar body volume, it's going to be 4/3 times pi, which they've told you to use as 3.14, times the radius cubed. So it's going to be this halved cubed. So 5.2 cubed. If we do that, we get 588.68, and then micrometers cubed. And then we need to work out the nucleus's volume, which is 4/3, 3.14, times its radius cubed, which is 3.5 cubed, which gives us 179.5. Therefore, the polar body volume minus the nuclear volume is the volume left that those mitochondria can enjoy. So 588.68 minus the 179.5 gives you 409.18 micrometers cubed. Now the question's saying how many of these can you squeeze in? So for every micrometer, you can squeeze 0.08 of them. It's saying that you can do 0.08, and per usually means over whatever, micrometers cubed. So every 1 micrometer cubed, it gives you this. So we have this many micrometers cubed. It's going to be 0.08 times 409.18. If we do that, we get 32.73, but we wanted to the same number of significant figures they gave us in the question, which we will go with 2. So it's 33. There you go. So now it says mitochondrial diseases are carried by faulty mitochondria. All of a person's mitochondria are inherited from their mother via the egg cell. An egg contains this many mitochondria. One proposed treatment to passing on faulty mitochondria involves removing the nucleus from an egg with healthy mitochondria, replacing this nucleus with the contents of the polar body from a woman whose egg cells are affected by mitochondrial disease, suggest how this will prevent the inheritance of mitochondrial diseases. So here, what we've got is we've taken our egg out with we've got a healthy one with a proper, lovely nucleus, and taken it out and replaced it with everything else from the other affected one minus the mitochondria. It's told you in the question that mitochondria are inherited from the mother. So if there's no faulty mitochondria left, then you're not going to inherit the mitochondrial disease. So immediately we can say that the egg created has healthy mitochondria, right? But the nucleus of the original parent or the nucleus of the mother, right? Therefore, there's less less inheritance of faulty mitochondria. Therefore, you'd have less mitochondrial, that's right, mitochond for short, disease inheritance. That's it. We're not producing healthy mitochondria. We're just getting, a cell that has healthy mitochondria, and there's going to be lots of normal ones rather than the faulty ones from the other one. That's it. If most of the mitochondria in a cell are faulty, this prevents many important enzyme catalyzed reactions taking place or slowing them down. Explain one reason why. So it's saying, why is mitochondria not worky mean enzyme not worky? So what that means is if mitochondria is not working, we're going to make much less ATP, right? So then we need to think about why that affects enzyme substrate thingy-majiggies. So it means there's less energy from ATP for enzyme reactions. That's all it wants. That's it. So binding of one molecule of oxygen to hemoglobin makes it easier for a second one to bind. Explain why. So oxygen binding to hemoglobin. Remember hemoglobin, right? It's a quaternary thing with like lots of different chains on and stuff, and you have an O2. And for some reason, all of a sudden, this one binding to it means it's easier for the rest. Now, if it's easier for the rest, the only explanation for that is that this must be more accessible to those O2s, meaning that this has to have changed shape. And that's exactly why that happens. It is the reason why the hemoglobin dissociation curve is like this. Because at the start, it's not very sharply increasing because there's barely any O2s. Then a couple bind, and then all of a sudden it's easier to bind, and bang, it starts going really sharp. At the top, it levels off, though, because remember, our saturation's really high. So if you think about it, HB here can have 4 of them, right? If there's only one free spot, most of them are filled, so it's a lot harder for the O2 to actually bind to it. So that's why it's that shape, and it's not just a straight line. So here, because if it didn't have that, it would either just be a straight line and level off like this, which it doesn't. It does that because of this. So then the reason why that happens is that clearly the binding of the first O2 must change the shape of hemoglobin. That means the further binding sites are more accessible or easy to bind to for O2. There you go. A hemocytometer is a special microscope slide which can count the number of blood cells in a healthy sample of blood. The surface of the slide has many small equal-sized squares marked on it. The depth of the liquid under each square is 0.1. So what it means is you've got these little squares, and underneath it, it's got a depth of that, right, 0.1. Now, when counting these, cells that touch the top and left lines are counted, but the cells that touch the right and bottom are not. The doctor used the hemocytometer to determine the number of red blood cells per millimeter cubed in a blood sample and then diluted it by a factor of 200 times before putting it down. Fine. Then it says, here's the distribution of the cells on a normal typical square. So remember that the depth of the liquid is 0.1, as in how deep this thing is, if we look at it side on. And they've told you that this has been diluted by a factor of 200 times. Its concentration has actually gone down by a factor of 200. This might be useful for later. The doctor counted the number of red blood cells in many different squares. The mean number was 7. The original blood sample was diluted by a factor of 200. Calculate the number of blood cells per millimeter cubed in the original thing. So there's a multi-step thing we've got to go on here. First of all, they've counted on this whole square 7, right, for means. So one square has a mean of 7, but they want it per millimeter cubed. So we need to work out the volume of this one square, right? So volume of a square is going to be its 0.05 times 0.05 times its depth, which they told you was 0.1. If we do that, we get 2.5 times 10 to the minus 4, right? So that's 7 in a volume of this. So we do 7 divided by 2.5 times 10 to the minus 4, and that will tell us the cells per millimeter cubed. So 7 divided by 2.5, so whatever, right? It gives you 28000 per millimeter cubed. But that's not the final answer, because this was in a 200 times diluted sample. So what we need to do is we need to undo that dilution. So you're going to do 28000 times by 200. Because remember, what we did to the original concentration is we times it by 1 over 200, so you could divide by 1 over 200, which is the same as saying times 200 over 1, which is times 200. So then if we do that, we end up getting 5.6 times 10 to the 6 red blood cells per millimeter cubed. That's how you do that. So then it says, when counting, cells that touch the top left lines are counted, but those that don't touch the bottom left are not. Suggest 2 reasons for this rule. So if we have a look, the ones that count the top left are, but the ones that aren't. I think it's just to, a 1. Imagine if you decided to count top left and others didn't, and others did and didn't, right? You're just going to get inconsistent results. So it's so that you can have 4 consistent results immediately. Because then, if you have consistent results, it means that every count can be relied on and can be compared because we're all following the same rules. The other thing as well, though, is let's have a look at what else we get here. Well, here you can see there's a part of a cell there and a part of a cell there. It's going to be really hard to gauge whether you would count it or where you wouldn't. Whereas if you just said everyone in the world has to count this and this, even if they touch, then you can kind of ignore that. And it doesn't matter if it's a part or it's a full because it's consistent. So then again, it's to avoid, counting parts of cells. It just means that you don't have to factor that in and say, oh, well, there's a half, there's a quarter. It's a bit subjective. So the doctor also wanted to know how many white blood cells there were in a different sample. To do this, they diluted it by 20 times, and then he made white blood cells become clear by using a stain that makes them appear dark blue. When counting the white blood cells, they only diluted it by a factor of 20 instead of 200, so just why. So if you're doing it by 200, remember, like if I have this here, right, this is my concentration, here's 2 cells here. If I then make this box 200 times bigger, they're way more spread out, right? So then if I decide to do it only by 20, they're going to be more spread out, but like they're not too far spread out. So the problem is if you do it really far, and then if I only look here, I'm missing out that entire other cell, whereas here I'm looking at the whole thing. So the reason that they didn't dilute it as much as and increase its volume too much is so that there are, enough, you can see enough cells, right? It's to see enough white blood cells, you would say the whole thing, obviously, like that, because there are fewer numbers of them. That's what the question's alluded to. There you go. Then it says, explain how this stain allowed them to count the white blood cells amongst all the red blood cells. Well, it just told you in the question that this stain, what does it do? It stains the nuclei blue. And it says, why can you do it with white blood cells versus red? Well, white blood cells have a nucleus. Do red blood cells have one? No, they do not. So red blood cells do not. They don't have any nuclei, so they won't stain. So scientists investigated the role of a protein called this in mitosis. They involved doing self-fractionation, which is separating debris and all that kind of rubbish as well and breaking them into chunks, and ultracentrifugation, which is where you're spinning it at high speeds and separating things based on how dense and how heavy they are, whether they settle at the bottom or not. The scientists began by lysing, breaking open cells and organelles, so they're breaking up these organelles as well using a detergent that dissolves lipids in water. Suggest how this detergent releases this. The question's told you that this is a protein involved in mitosis, and it's also told you that we're breaking open cells and the organelles using some mysterious detergent that dissolves lipids in water. So clearly, lipids breaking is somehow leading to these cells breaking open and protein leaking out. So the only lipids that would be stopping them from doing that, remember, is your phospholipids. So what the detergent does is the detergent must be dissolving phospholipid bilayers, right? Which make up cell membranes. That's it. Got to work with what the question's telling you here. So now it says, explain how ultracentrifugation separates this from other molecules. So here, this is a protein. Ultracentrifugation is where we're spinning loads and loads and loads of things and then separating them based on density. So what you're going to do is you're going to spin the sample at increasing speeds because you're going to make them faster and faster. And you separate the CENPW based on its density or mass. But we're not done though, because this is a protein and it wants it from other molecules. Because it's a protein, we need to spin it. So, this would only separate. So CENPW would only separate at very high speeds because it's not very dense. That's why. Because it's not as dense as, like organelles and other stuff. And proteins, etc. There you go. So CENPW is involved in the formation of spindle fibers in mitosis. Spindle fibers are made from molecules of a protein called tubulin. The scientists retreated the cells in a culture with small interfering RNA. All right, this siRNA causes RNA interference of expression of the CENPW gene. They then took samples of cells at 0, 48, and 72 hours after adding this siRNA, and then they used gel electrophoresis to separate the CENPW and tubulin from these samples. So here's the results of the electrophoresis. The size of each band is proportional. So the size of each band is proportional to the amount of this in the tubulin present. So it says, suggest how siRNA produced these results. So we need to go back to the context of what the question's saying. So here, it's told you that they've, again, used siRNA, and what it does is it causes RNA interference with the expression of this. When you want to express a gene, you need to transcribe it into mRNA, and then you need to translate it, right? So the mRNA becomes a protein. That's how all of this topic works. So here it's told you that for some reason siRNA causes RNA interference, as in something is not letting this thing happen. What siRNA is you have double-stranded RNA. It's called small interfering RNA. And what it does is it carries a little protein associated with it as well, right? So what I mean by that, siRNA is double-stranded small interfering RNA, right? What happens is it gets into the body, whatever, and then it splits. One of them is preferred, it's selected. The other one kind of just gets broken down, right? And what it will do is it will associate to a whole bunch of proteins that are going to eventually break things down. This siRNA then forms complementary base pairs with the mRNA of whatever you want to interfere, as in here. Then what it will do is, because now these base pairs have happened, there's all these other proteins that are now stuck with this one strand that now breaks this into fragments and chunks, and then it's no longer translated, so you're not expressing it anymore. So here, it's saying, suggest how it led to these results. Well, we need to talk about the first steps. So what it does is that this siRNA binds to this mRNA, because that's what we're making, remember? So CENPW mRNA. So it's not translated. but that doesn't explain how we get to the results, right? So can you see how they've done it after hours, after time? They've mixed them together, they left it for a bit and left them over time. Can you see how this drops? It starts here, it drops, this here, start and drops and drops and drops. So clearly, over time, it's decreasing. But in the question, it's also told you that CNPW is involved in the formation of spindle fibers. Spindle fibers are made of tubulin, so both of them are dropping. So this means that over time, CNPW decreases, and since spindle fibers are made of tubulin, you're also going to get a decrease in tubulin over time. That's all it is. Got to use the question. So ammonia in soil is oxidized to nitrates and nitrates by nitrifying bacteria. Scientists investigated whether two soils with a different pH contain different communities of nitrifying bacteria. These communities all consist of these nitrifying bacteria of different species in each soil. They took samples of the soil from two sites, A and B, and they measured the pH and found that this had a pH of this and this had a pH of this, whatever. They measured the concentration of ammonia soil samples over 20 days, and each sample contained the same number of ammonia at the start and had the same mass. They recorded the concentration of ammonia in A with a pH of this, B with a pH of this, and a mixture of equal amounts of both with its pH adjusted there. And here's their results. So immediately, number one is to work out its independent and dependent variables, right? So independent, dependent. So what it is it's going to be soil pH that they're changing, and the dependent is the concentration of ammonia, right? Now, if we have a quick look at how this all links again, remember, what they're trying to do is they're trying to look at the soils if having different communities of nitrifying bacteria, and they're trying to look at whether the changes in ammonia being made is a surrogate for that, as can this represent this? And they're trying to see whether the pH is causing changes in those as well by measuring this, if that makes sense. Now, they've established in the question that these communities in these soils have different species. So each of these soils have different species in them. Fine. If we have a look at the data, so concentration of ammonia is dropping over time. That makes sense because remember in the question, it's told you that it's oxidized into nitrites and nitrates. So you'd expect it to go down. So if we have a look at A, let's look at B, sorry. So if we look at B, it drops quite quickly and then it sort of levels off. So what must be, and this is in a pH of 4.3, whereas in A, it's dropping kind of slowly compared to this, but it starts to decrease and it levels off a little bit earlier really than B does, because B really levels off about there and it stops here. Whereas when you mix the two of them together, it seems to drop the slowest and then levels off roughly at the same point as where A is. Fine. The scientists use this concentration of micrograms per gram for the concentration of ammonia in the soil, suggest why they use these units. So if we look at the microgram parts, why would they have used micrograms? Micrograms are teeny, so it must have been that there are little amounts of ammonia in the soil. It wouldn't make sense to just deal with 0.0000 blah, blah, blah. It'd be easier to just change the units so that you can see the numbers a bit nicely. So the reason is that because it then means you can avoid having to use, like I just said, lots of decimal places. Or you could say standard form, right? That's that bit there. What's the per gram bit mean though? Here, it's the concentration of ammonia in the soil, which is the micrograms of ammonia per gram of soil, because that's what it is. So this means it's to allow comparison between the samples. Because, for example, if I look at humans, right, and let's say for some reason I have these other beings that only have legs, no arms, some mysterious other beings, and I have 3 of these, right? You can see that per person there are 0 arms per being. Whereas here, even though I have 2 people, we have 2 arms per human, right? Even though I have 3 here and 2 here, these are comparable numbers because each human has 2, each mysterious being has 0. If I had 1 million of these and they all did that, it's easier to compare than by saying, well, I had like 200 of these and they all had no arms, and I have 10 of these and they all have 2 arms. Like, this gives you a comparable number, which is why the whole per thing is done. So there you go. So then it says calculate the difference in the rate of breakdown in ammonia per day on day 0 and day 2 in soil A and B and give you units. So here, this is showing you the concentration of ammonia, right? And this is time. The rate is the change in the concentration of ammonia, or the rate of anything generally is over time, right? Now, if you have a look, the change in Y, right? We can rewrite that as the change in Y because that's our Y-axis. We can change this to change in X because that's our change in X-axis, right? Change in Y over change in X. Does that look familiar? That is your woohoo! It's your gradient. Wow. So we're going to work out our gradient. So for A, we're going to work out the gradient, and for B, we're going to work out the gradient too. So the gradient is Y2 minus Y1 over X2 minus X1, and for the same for this as well. So Y2 minus Y1 over X2 minus X1, and we're doing it from day 0 to 2. So look for where day 0 to 2 is. So then if I read this graph here, I've just done a quick skipped ahead a little bit. We've got that one there. That's for our A. This is for our B. So our Y2 for B. Sorry, for our A, my Y2, here was 14, and then the original was 20. So it's going to be 40 minus 20 over the time frame in which I did it, which is 1.25. I chose this. It's anything before 2 works because the line is consistent, the gradient is consistent, right? So then for B, it's going to be this point here, which is 10 That's 8.5 minus 20 over the same time frame. So 1.25 minus 0. So if I work out the gradients, I get minus 4.8. And for the second one, we'll get minus 9.2. So then the difference in rate is just going to be minus 9.2 minus 4.8, which gives us 4.4. There you go. So the scientists concluded that the soil mixture experiment showed there were different communities of bacteria in A and B. What evidence supports their conclusions? Give reasons for your answer. So it's only talking about supporting it. So the questions told you that we have ammonia is oxidized to nitrites and nitrates by species of nitrifying bacteria. So you have differing amounts of changes in ammonia, then you're going to have differing amounts of this and this, which are done by different species. So clearly, because there's differences in these, already we can say that there's some differences here. If we look at if you look at B, I keep going to A because I keep thinking it's that, sorry, whoops. If you go to B, you can see that this line is most steep, it's dropping the fastest, it's having the fastest rate of breakdown, right? Whereas if you look at A, it's a lot slower. So it's implying that these changes are due to their pH. So it implies that there's different species within them, right? But when you mix the two of them together, they're both doing rubbish. The rate is just kind of crap. So it implies that there are some bacteria here that prefer to be at 6.9 and then some that prefer to be at 4.3. When you put both of them in the same pH here together, well, here, B doesn't like being at 6.9, so clearly it's not working. And A, although being in the same pH, 6.9, for some reason being mixed with B doesn't seem to work, as in, it doesn't seem to be very happy. It wants to just be A on its own. So clearly, the fact that these two mix together means it's not working very well, means that there's different communities. Otherwise, if it was just A and A, they're the same community, then it would be this equally as good. Or if it was just B and B, then it would just be rubbish. And it would just kind of do this much more rubbishly. So that's what we're going to say. So B has the fastest decrease in ammonia. I'm just going to because I'm lazy, but you do the whole thing. Concentration, right? Whereas A plus B is the slowest. So what this does is this implies there are different species of bacteria in A because the pH is the same, A and B, sorry. The pH is the same for A, but mixed together, the overall rate decreases. So clearly, there's something, there's a bunch of them preferring different pHs. Therefore, the bacteria don't work as well in this pH mixed together. So the oxidation of ammonia by nitrifying bacteria involves this enzyme ammonium monooxygenase. Each species of nitrifying bacteria has its own specific AQA gene that codes for the production of ammonia monooxygenase. So fine, each species of nitrifying bacteria has its own specific AQA gene that makes this, right? So scribble that down. AQA gene leads to I'm going to just call this ammonia oxidase, right? Just going to call that ammo-oxidase. Just something to know that I know what that means, right? So this codes for this production. And what else did we know? What this does is it will oxidize ammonia into nitrites and nitrates from the whole question, remember? And it's also told you that each species has a slightly different version of this. Fine. In a second investigation, the scientists determined the expression of this gene with two species of bacteria, S and T. S was from acid soil, and T was from a neutral pH. So they drew cultures of each one of these, and then they measured the amount of mRNA from the AQA gene in each culture. So again, dependent, independent. The independent is pH. The dependent is mRNA copies, right? But the mRNA copies are of this AQA gene, which remember each species has different versions of. Cool. So we have a quick look. So species S in acid soil, as you can see, makes more mRNA copies of the AQA gene in acid soil, and it drops as the pH goes higher, it becomes less acid. Whereas T, when you put it in acid soil, has less mRNA copies of those AQA genes, but it starts to climb up as you tend towards the neutral region there, which is 7. Which species was the number of mRNA copies more affected by changes in soil from this pH of 4.9 to 4.75? Use a calculation to support your answer. So what that means is if we compare 4.9 to 7.5, we're looking at the difference between them. So for S and T, work out the 4.9 copies and then the 7.5 copies. So if you look at S here, it's 10 to the 6, right? And then if we look at T, it was 10 to the power of 2, right? Whereas at the 7.5, they were both 10 to the 4, okay? So then what we do is we work out the difference. The difference here is going to be 10 to the 6 minus 10 to the 4, which gives you 9.9 times 10 to the 5, whereas 10 to the 2 minus 10 to 4 would give you minus 9.9 times 10 to the 3. Now that doesn't matter because it's just asking what the changes or the difference between them two, right? Just the number. This will have dropped by that much and that's going to drop as we went up, which is why one's negative, one's not, right? So here it's S because it changes by 9.9 times 10 to the 5 versus T, which is only 9.9 times 10 to the 3. There you go. This method allows a scientist to estimate the expression of the MOA gene in each culture, but not the growth of the bacterial population. Explain why. So this question again, let's go back to the thing here. So it's saying it could estimate expression of MOA, but not the growth. So we need to look back on this and think what's up. So why can't we just conclude, essentially is the question, why can't we just say, oh, they're growing more, right? If you have a look, this is only measuring the copies of mRNA. That doesn't tell us how many mRNAs are being made by how many cells. We don't know if one cell's making loads or one's making one, right? You can't assume that one mRNA is made by every cell. When you're making a protein, for example, if you want to make salivary amylase, your amylase, your like glands that are going to make them are not making one mRNA, because that would only make one amylase, and that's tiny, and you would barely digest anything. We're making bajillions. So we don't actually know how many cells are making this. We also, they're saying we can't use it to estimate growth. All this does is it tells us about, and this is why I wrote this here, all it does is it converts ammonia eventually into nitrates and nitrites. It does not tell us anything to do with cell growth in this case, which is their cell division, which is binary fission. It has no link there. So that's why we have to explain why. So in these kinds of questions, you need to think, is what they're measuring actually directly relatable to the thing that they're trying to claim? In this answer, no. So, and as well as that, sorry, I missed the most obvious, we don't actually have the number of bacteria either. So we have no number of bacteria. Also, we do not know how many cells are making copies, making how many copies, as we don't know the number of copies per cell of mRNA. Not even just that, because as well as that, we don't actually have, we need to say why that's relevant. This only tells us how much transcription is happening. We don't even know how much translation is happening because we don't know. We haven't measured the protein. No amount of protein, so we don't know the amount of translation happening, right? But then as well as that, again, we only know that this is linked to ammonia stuff. We don't know if this AMOA, RNA and protein, is linked to binary fission. That's why you can't just jump ahead and say that. Like with my chocolate shitting example I gave before, where 10 people eat chocolate and they shit themselves, and I say, oh, that must mean that these people are lactose intolerant. You can't conclude that because I gave them chocolate, and I have no idea whether it was lactose that did that, or whether it was the ingredients in the chocolate, or maybe I don't know, gave them I just had really unhygienic hands and gave them hepatitis A and diarrhea and all kinds of stuff like that. So you don't know, Scientists set up their cultures in sterile glass bottles to suggest one suitable method for sterilizing the bottles and explain why it's needed to do that. So these are bottles. So you're not going to flame the bottle. You're not just going to heat. It's so, you need to say what you're going to do to a bottle. So what you do is you would, you could, for example, wash the bottle in disinfectant, or you could autoclave it, which is where you heat them really, 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 really hot, or you could heat them wash them in steam or alcohol or something like that. And the reason is because we need to remove other bacteria, but that's not enough. Because when you're doing these questions on experiments, and when they're saying they did this, say why this is important, you need to think, how is it going to screw up this dependent variable? All of these things that we do, these controls, is to make sure the only thing that changes this is the pH. The only thing that changes the dependent is the independent. So you need to think in your head, why would removing bacteria stop this being screwed around with? It's because there are other bacteria, and we're looking at how much ammonia is being broken down and ammo that's being produced, right? So we'd remove other bacteria that could produce AMOA, or you could say that could break down ammonia, which remember would screw up the results from the other question from before. That's why we do those controls. It's not just to allow comparison. That doesn't mean anything. It's to make sure that the thing you're changing is the only reason the thing that you're measuring is changing. So here, this is the dreaded essay that everyone's really worried about, and it's reasonable to be worried about it, but you should come in with a strategy. So a lot of people say you do this and do that, whatever, fine. I think a good thing that you could do, and I think other people have mentioned this, is read the essay at the start, go back and do questions, and you'll be thinking about it throughout. But when you're doing the essay, try to be able to plan what you're going to bring up in 5 minutes, I think, if you can. And then spend the other 25 minutes doing this, right? You can spend a bit longer if you want, but if you practice at that 25 thing, you'll force yourself to work faster and faster is what I think, right? But obviously, as they say, don't spend more than 45 minutes on that. Please do not spend ages on this. The payoff is rubbish. Now, the rules are here is everyone loves to try and itch to try and say the off-the-spec stuff for the extra marks. That's 5 extra marks. And every exam report, if you read them, says all kinds of stuff. In one exam report in 2019 or 22, whichever one it was, some people tried, remember that about 20,000 people did this exam. Enough people tried to go off the spec, quote unquote, by mentioning the water cycle, right? But the problem is that the depth they set it at was at the same level as key stage 2, as in year 3 to year 6, as in you are 8 to 11 years old. It doesn't work. You can't just hope to say something else and you will get the marks. In the same sense, you can't just jump in and say menstrual cycle at the GCSE level as well. You need to say this if you're going to do that at A-level standard. But the other problem, though, is if you say something that's significantly wrong, that can take you out of a certain banding of marks and lock you out. So I think if I have a look at the mark scheme 1 sec, if you have perhaps one significant error or one irrelevant topic, then you will get into relational or integrated as a whole, which is a maximum of 16 to 20-ish marks, right? Whereas if you decide to get all cocky and be like, yep, I'm going to go for the big boy marks, and you say one thing that's irrelevant, but then you accidentally now screw yourself over saying another irrelevant thing, now you are stuck at 11 to 15, even if everything else is pristine. So I personally don't think that it's worth trying to go for that extra spec stuff, for the maximum of what, like 4 or 5 marks? If you really know what you're saying, then fine. And if you can say it in enough detail at A-level standard, then fine. But I just don't think it's worth it. So the exact essay that I chose during my A-levels was this one here, because I just thought it would have been way nicer to do. But both of them are still quite nice. I'm going to talk about the one that I did, and I'll show you what my strategy was, right? Whenever it says importance, you can't just jump in and start talking about like a whole monologue of how this works, recall memory stuff, and then at the end say, this is important because we will die. It's useless. Don't do that. Examiner reports, every single one of them complain about you guys doing that. It don't work. The importance is why this thing works, why this thing happens, why each and individual little step is important. So if I'm looking at interactions between cells and organisms, what I literally do is I just think of every single topic in my head. 1, 2, 3, 4, 5, 6, 7, 8. And what I do is I go through them one by one and think. So topic 1 is about biological molecules, right? So, DNA, ATP, proteins, lipids, all that kind of stuff, right? That's not really anything to do with between cells and between organisms, so you're out. Topic 2, well, if we think about it, we're talking about cell membranes, right? Cell membranes, interactions between cells, they have receptors. So we can talk about cell membrane receptors here, right? In your head, as you're doing that, what you're trying to do is you're trying to pinpoint the exact important thing. Don't just write the whole topic. That's useless. If I say cell membranes, that means nothing. What I'm doing is I'm knuckling down on what the really important thing is here, right? So that's that one. But if we talk about topic 2 as well, that's all to do with the immune response. Hello? That's pretty dang important, right? So rather than just saying immune response, narrow it down to what it is. So obviously, the entirety of the immune response is actually it, to be honest. But then even more specifically, antigen presentation, right? Chemical signals, and also replication of viruses as well. When they get into a cell and do all of that jazz. That's pretty dang important, is it not? So then, moving on, that's all I can think of for there. The next topic is all about exchange and transport systems, right? So gas exchange and this and that. So if we're talking about interactions between cells, if I have a really strong think about it, what can I think of here? Well, we have capillary cells and, alveoli interacting with each other in human gas exchange. That's important because, diffusion across and stuff like that. So capillary alveoli. In leaves, you also have your stomata as well, which is quite important. So another important thing is your flow and your mass flow hypothesis. That's pretty important. That's involving cells and your companion cells and this is that. That's that. So then topic 4, if we have a thing, that's all about genetic diversity and classification and DNA replication and all that jazz, right? And natural selection. Between organisms, we have courtship behaviors. That's important because then you're able to recognize the members of the right species and this and that, blah, 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 reproduce and pass on your thingamabobs. That's pretty important. As well as that, interactions between them. You've got predator-prey relationships as well and things like that too, which affects biodiversity and stuff as well. So what I mean by that is for example, if you have all of the same species and they're all the exact same, one virus will wipe them all out. Whereas if they're reproducing and this and that and interacting and interbreeding with lots of different populations, there's good genetic diversity, so they won't die and get wiped out by this one thing because there's genetic variability. There's alleles of different things, so one might be allowed to survive and reproduce and this and that. Right, there's that one. Now, if you have a look at number 5, that's photosynthesis and respiration. Not really much there. Nutrient cycles is pretty important. Nitrogen cycle involves things between organisms, like plants taking up nitrates and ammonia and stuff like that as well. Nitrifying bacteria and all that stuff. And on the same vein, the phosphate cycle too. That's pretty important. This topic's juicy as hell because you've got all of your receptors. You've got your Paxinian corpuscles, you've got your photoreceptors, and you've got your control of heart rate. Those are all gigantically important things, right? So those nervous impulses there. And as well as that, you've also got muscular contraction as well. It's pretty dang big. And not even just that. You've got the control of blood glucose. Insulin, remember, is released from beta cells and it goes through the blood. It binds to receptors on skeletal and liver cell muscles and it causes GLUT4 to come up. That's pretty important. And glucagon as well. And also control of water potential in the blood as well. For example, osmoreceptors, they release impulses to the pituitary gland, which releases ADH, and it travels through the blood. It binds to collecting duct cells, and then it puts aquaporins there, and it means you can increase the reabsorption of water, make less urine, so it means you keep the water in your body, so the water potential is right, so your cell body processes can carry on. That's pretty important, because there's interactions, the receptor bindy bits, right? Other bits you could talk about as well are evolution and speciation. So like, for example, if you had, the reason that we exist today is the fact that these things couldn't interact with each other, so that we had speciation because of the reproductive isolation, as well as that your predator-prey relationships, which I mentioned up. And there's some pretty hefty ones, as well as that you could probably tie into cancer and stuff. So what I would do is when I'm saying the importance, if we take like, for example, the immune response, you don't just list a whole monologue, you talk about in between. So I usually start off with an example of the interactions between cells and its importance is the immune response. When a pathogen enters a body, phagocytes are attracted to this pathogen, engulf and digest it, and then with the lysosome, all that stuff, right? And then it will present it to a helper T cell by putting the antigens on its cell surface membrane. The helper T cell has complementary receptors that bind to this pathogen, and this stimulates the helper T cell to release chemical signals, which stimulates cytotoxic T cells that kill infected cells, more phagocytosis in B cells to undergo clonal selection and make monoclonal antibodies. This is important because it notifies, obviously you're paraphrasing here, you'd say in a proper biology way. This is important because it allows the body to mount an immune response and recognize a pathogen in the body. Otherwise, the pathogen will be able to replicate without the immune response being able to mount an effective response, which can cause disease, illness, cell destruction, and death. That's why it's important. You're really pinpointing the importance on the receptor bit being there. Right? A lot of people just write a whole monologue and say, this is important because if you look at the controller heart rate, baroreceptors detect a really high blood pressure. For example, they'll send impulses to the medulla via sensory neurons, and then what will happen is the medulla will send impulses to the sinoatrial node via parasympathetic nervous system pathway. ACH is released, which binds to receptors on the sinoatrial node. These interactions are really important because we need to lower the blood pressure because it can cause damage to the heart, it can cause damage to blood vessels, and can cause disease. And it allows us to have that control because the other side happens as well. When your blood pressure is too low, you have a noradrenaline. And it's those interactions you have to highlight. The impulses getting to the medulla means that the thing is, the low blood pressure is actually processed into a response, and then the impulse is going from the medulla to the receptor means that the effector can actually carry out something. Without this, we'd have no control over our heart rate, which means one, it'd be too high, for example, the blood pressure, and it'd cause damage to the vessels and cause death. And two, it wouldn't allow for fight or flight responses. For example, if you wanted to have more blood getting to muscles for respiration, for more energy, so there's muscle contraction getting by. Usually I'd say pick about 4 topics as best as you can and make sure they are synoptic and they are linked and they have like a really good hefty thing. So maybe you could start by introducing the concept of receptors being the means of interactions between cells. Cells interact with each other generally in your blood by hormones floating around, right, and binding from one to another, or they interact, for example, a pathogen binding and getting into something like that. So it's really important you identify the importance, not just list out a whole monologue and then say, here you go, that's what it is. So I always say, quickly scribble out like a plan of 4 juicy topics that you can think of in your head and really pinpoint what it is and why. Courtship behaviors between organisms is important, so they recognize members of the same species and also when mating season is due, so that they're able to reproduce and continue passing on their genetic material so the species doesn't become extinct. Can you see how that's really important, where I've narrowed it down on that one thing, not just listed the entire process out? Anyway, I hope that helped. Please like, share, and subscribe. You know the drill, and thank you very much.